So thank you for the introduction. I'd like to thank the Covertly Foundation and the organizers of the session for inviting me to speak. So this is the deep brain stimulation session, and I am going to talk a bit about deep brain stimulation. But really what I'm going to talk about is DBS, and in the context of what we can learn about how the brain works just by the fact that DBS now exists. So how do we study the brain in living people? Well, you can do really clever behavioral experiments like Dr. Frank does, and sometimes you don't even need technology. You just need the behavior to make some inferences about how the brain works. You can do things the old-fashioned way, which was to wait for someone to have a neurologic problem, then wait for them to die, and then cut up their brain and see which part of their brain was messed up. But that's an incredibly morbid and inefficient way to do neuroscience. So we have better tools now, so we can do things like take pictures of the brain. So we can do MRI scans, we can do PET scans, image molecules within the brain. Those are the types of things that give us information on long time scales, at least hours, probably days, months, years. We can do EEG, so we can put electrodes on the surface of the scalp. We can record the cortical potentials underneath. We can do things like diagnose seizures. We can do behavioral experiments while people are being recorded. As an aside, this technique was invented by a Prussian soldier who believed that he could communicate telepathically with his sister several hundred miles away. He devoted the rest of his life to trying to figure out how that works, so we have him to thank for that. And then the other thing you can do is you can put your electrodes right up next to the neural tissue, right up next to the neurons that you're trying to study. This is obviously a problem in human subjects. And as you go from these sort of bigger scale, longer time scales down to the very short time scale, you go from less invasive to more invasive. And you also go from bigger to smaller. So you go from taking pictures of the entire brain or recording from the entire cortex to potentially recording single action potentials from single neurons, or maybe even recording synaptic potentials. And that's what I'm going to look at today. So we saw in the introduction some images of Parkinson's disease, so I'm not going to belabor the point. But this is a patient with Parkinson's. She moves slowly. Her face isn't particularly expressive. This was originally described by Dr. Parkinson in 1817. For those of you that like these sorts of things, you can actually download this from iTunes and read it in the dentist's office while you're waiting. And Dr. Parkinson's description was really, really good, even though it turns out that some of the patients he described probably didn't actually have Parkinson's disease. So the title of my talk is about the basal ganglia. So why do we care about the basal ganglia in Parkinson's? Well, as Dr. Hamada mentioned, there's this chunk of brain in the middle, and down in the midbrain, there are these dopaminergic neurons in the substantia nigra. Those neurons die in Parkinson's disease, and those neurons send dopamine primarily up into the striatum, although to other places as well. And this was recognized in about the 50s, that the striatum is particularly dopamine deficient. By the 1970s, the chemists had figured out how to replace brain dopamine levels with a pill. And so we can be reasonably certain that dopamine is in fact the culprit, at least in the motor symptoms of Parkinson's, because we give dopamine back and patients go from moving like she did earlier to moving like she is in this video. So that's great. The problem is that as Parkinson's disease progresses, you go from moving too little to moving the right amount on medication to moving too much when you get your medication. And it gets very, very difficult to take patients and keep them in that happy zone where they're moving, but they're not moving so much. So when you see Michael J. Fox on TV, what you're seeing is the side effects of his medication. You almost never see him off, because Parkinson's patients would much rather be moving uncontrollably than not moving at all, as a general rule. So that brings us to deep brain stimulation. So this is a treatment for patients who have gotten to the point where we really can't control their symptoms with medication. And so to explain that, I'm going to allow our celebrity doctor in the audience to explain it. So if you don't know, The Doctors is a syndicated talk show on where they've got a panel of 4 physicians, and they'll bring up different medical problems and discuss them and give examples. So this is a patient that got her DBS at our medical center. I was a fellow at the time. No one told me that this was happening, so I'm nowhere to be found in the video. I don't think that's a coincidence.
So the next step is going to be the microelectrode recording.
As we pass it into the brain, we can hear sounds of individual. Today we're going to be performing an operation called deep brain stimulation. The deep brain stimulator is really a brain pacemaker. It's an electrode that's placed within the brain that synchronizes the signals so that these bad symptoms are overwhelmed by a positive electrical pulse. She'll be awake during the entire procedure so that we can do testing. Once the MRI is done, I do the mapping and selection of the precise target in the brain. Okay, we're going to get started. Good. To place the deep brain stimulating electrode, we need to make a small hole in the skull.
Right. So I just want to point out that this is a very neurosurgeon thing to say. We're going to make a hole in your skull now, like this is what you do every day, because it is what you do every day. And to the rest of us, that's a very bizarre thing to have someone say behind your head.
How's it going over there?
I'll be better when this is over, but I'm not feeling any pain at all.
So the next step is going to be the microelectrode recording.
As we pass it into the brain, we can hear sounds of individual cells. The shape of those individual spikes can confirm that in fact we're in the region that we expect to be. So I'm now getting the DBS electrode that's now in the brain.
So I'm going to stop there because that is the main point I wanted to make, that we have this wonderful opportunity during deep brain stimulation surgery to go in and actually record single neurons from awake, living human patients. And that can give us a lot of information. So before we move on to what people have seen when they make these recordings, let's talk a little bit about what you can record with a microelectrode. So there we go. So this is an old figure from a review about how these types of microelectrode recordings are made. So you take your microwires, in this case a bundle of microwires, you drop them inside the brain, there's a bunch of neurons around them. Those neurons generate currents. So ions flow in, ions flow out. This creates dipoles. Those dipoles generate electric fields. You can detect those at the microwires. When you stick this in there, what you get is a signal that looks kind of like this. You have these sort of slow, ongoing fluctuations, and then superimposed on that, you have these big spiky things. And you can do some pretty simple processing. So you can high-pass filter this, draw a threshold across here, and you can pull out the times at which all these spikes occurred. And these are the action potentials measured extracellularly that are sort of the information currency of the brain. And you can low-pass filter it, and you can get these local field potentials out. So local field potential is sort of like a depth EEG, and it's a little bit harder to interpret physiologically than the individual spiking. So this is believed to represent sort of the subthreshold transmembrane fluctuations in transmembrane voltage that are going on sort of collectively among all these neurons. All right, so what happens during deep brain stimulation surgery is the surgeon makes their burr hole, they drop their microelectrode in, and they start recording. And each of these pieces of brain sounds a little bit different when you listen to it on that amplifier. So the thalamus tends to have these very bursty units. As you get into the subthalamic nucleus, which in this case is our target, it's not the only DBS target, it sort of sounds like rain on the roof. You have this very loud, like thunderstorm, patter, 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 patter going on. And so that allows us to know when the electrode's in the right spot. And it tells Dr. Patil that this is where he should go and put the big electrode in that's going to actually do the work of the stimulation. But of course, what we can do all along this trajectory now is we can make recordings, and we can even potentially have patients do simple tasks during DBS surgery. So what do we see in Parkinson's disease? Well, pretty much no matter where we put the electrode, so this is sort of a standard box and arrow model of the basal ganglia, where each of these boxes represents a different anatomic region. What you see is that the neurons tend to fire in these brief bursts, so they fire a lot and then they pause, fire a lot and then they pause. And not only do they do that, but they tend to do it in a fairly periodic manner. So if you plotted the firing rate of these, it would go up and down in an oscillatory manner. We have a problem with these recordings, though, and that's that if we're recording from Parkinson's patients, we don't really know what normal brain looks like. And so there's a couple of ways that we can attack that. So one way is we can resort to our field potential recordings, which are much easier and much more stable. And we can give the patients levodopa. We can give them dopamine replacement, and the patients start moving better. And these sort of relatively low frequency, high amplitude oscillations that are going on in the subthalamic nucleus, they go away and they're replaced by these relatively lower amplitude, high frequency oscillations. And that's a very consistent finding across Parkinson's disease and its animal models. So there's another thing that we can look at, and that is we can look at the relationship between the field potential and these individual action potentials. So this is a filtered field potential from the subthalamic nucleus. These are when single neurons are firing. What you can see is it looks like there's a relationship. These neurons tend to like to fire at peaks of the field potential. And you can quantify that by taking out little snips of the field potential around each spike and averaging them all together. So if the spikes don't care about where you are in the phase of the field potential, you should get a flat average. And if they do care, you should see an oscillation. And you see a large number of units that are locked to this field potential. So this implies that there's some sort of widespread synchrony going on in the brain in Parkinson's disease. Now, as I mentioned, we don't really have controls for human patients. We can use them as their own internal control after we give them levodopa or turn on the DBS. But we do have pretty good animal models of Parkinson's disease as well. Not great animal models, but pretty good. And especially the toxin models. So we have drugs that will poison dopamine neurons, but not poison the other neurons. So we can give those to our rats or our monkeys, kill off dopamine neurons. So this is rat striatum, and this is a stain for the dopamine terminals. So here a lot of the dopamine terminals have been killed. And what you see in all of these animal models, pretty much wherever you look, that you get this oscillatory pattern of bursty activity. And you get a lot of neurons that are locked to the field potential with this big spike-triggered average. And you don't really see that in the healthy animals. So I'm cheating a little bit here because these are clean signals from an anesthetized animal, but it turns out that you see the same thing in awake-behaving animals too. So what this suggests is that in Parkinson's disease, a substantial portion of the pathophysiology is related to this increase in neuronal bursting, this increase in neuronal oscillations, and this increase in neuronal synchrony. The question then is, all right, what causes these changes, for one thing, and then how do these changes cause Parkinson's? Do these changes cause Parkinson's? Or which of these changes are directly responsible for causing the symptoms that we see in patients? So that's really hard to answer in patients because we have such a limited capacity for recordings, although the recordings do at least, they really help us validate our animal models. So these are some experiments that we did. So we have a rat. This rat is doing a task. It has this satellite dish on his head. The satellite dish has a bunch of microwires in it that go down through a hole in its skull into its brain. And we can continuously record neural activity in a lot of different brain regions. So down here, we're looking at the power spectrum, sort of the rolling power spectrum of the field potential. And up here, these are individual neurons spiking. So in this case, the rat is supposed to poke its nose into one of these lit ports. It gets a cue. The cue tells it which direction to go. If it does everything right, it gets a sugar pellet at the other end. So we were actually doing these experiments to try and test hypotheses about some of these issues of impulsivity that Dr. Frank was talking about yesterday. And for that reason, we were recording from the subthalamic nucleus, which he was talking about yesterday. These are healthy rats, by the way. These are not dopamine-depleted rats. But what we noticed in the course of these recordings, we were recording from all these different regions, and there were these little episodes lasting a few hundred milliseconds, maybe 4 cycles, where the entire basal ganglia would oscillate together. So the parts obviously highlighted in red here. And then each region would sort of go back to doing what it was doing, and then they would oscillate again together. And it turns out that this is highly predictable by what the animal's doing at that time. So there are certain events in this task where the basal ganglia really coordinate their activity, and there are certain events in this task where they seem to be working fairly independently of each other. So what I want to show you now is a slightly different measure of that spike field synchrony that I showed you earlier. So if you have a sine wave, and you have, say, this black neuron here, and this black neuron does not care at all about the phase of the ongoing oscillation. And then you plot a histogram of when those spikes occur with respect to the phase of the ongoing oscillation, it's basically flat. If you have this red unit that really likes to fire at the peak of these action or at the peak of the field potential, you should see something that looks like this big peaky histogram. And when we look at the units that we recorded across the basal ganglia, so in all 4 of these brain regions that are really far apart in the rat, we see a lot of units with this pattern where this histogram is peaked. And you can do some statistics on it and say, well, how likely is it that this really is a flat histogram that by chance alone we missed? And usually the numbers turn out to be really small. And if we count up the number of units that are indeed locked to these beta oscillations, these ongoing oscillations that look a lot like what goes on continuously in a Parkinson's brain, we see that a lot more of them are locked to the field potential than we would expect by chance alone. So that's kind of interesting. But we also found a lot of units, and I think this is really cool and also very hard to quantify. We also found a lot of units that really didn't seem to care about what the local field potential was doing over the entire task. But if you looked at specific behavioral events, so like just when he's pulling his nose out, just when he's poking his nose in, all of a sudden these units really seem to care about what the field potential was doing. The problem is that this is a huge multiple comparison problem, so it's very hard to say that this is significant in a global sense. But it's certainly suggestive that what may be happening is that you have ensembles of neurons across the brain that are transiently locking into these rhythms, and that's a way for something that's way back here, like the subthalamic nucleus, to coordinate activity with the brain region way up here, like the striatum or even the cerebral cortex, so that if the field potential here and here are locked to each other, and neurons here are locked to this field potential, that means this neuron is also locked to this field potential. And that may be a way for regulating timing and communication across brain regions. So this is not a unique idea to Parkinson's. This has been studied extensively in a lot of diseases. There's a lot of open questions about what's causing what, but it's an interesting solution to what's called the binding problem of how does the brain integrate lots of data all at once. So getting back to Parkinson's disease, so what we can say from our experiments, beta is this transient basal ganglia-wide state that it sort of flits into, flits out of, and that seems to be in a behavior-dependent manner. And that individual neurons like to entrain to this beta state, and then they'll leave the beta state. And it may be that there are certain ensembles that entrain at different points in behavior to regulate what the animal's doing. And this seems to persist after dopamine loss. So you suck the dopamine out of a brain, and all of a sudden this beta keeps going and going and going. So what does that have to do with Parkinson's? So unfortunately, this green doesn't show up super well. But so each of the dots in this top part, this is a figure from a review by Peter Brown, who's a big investigator in this field. Each of these dots represents a neuron firing. And so if you have a normal amount of dopamine in your brain, your field potentials look reasonably normal, and these neurons aren't particularly synchronized, things go on. And as you suck the dopamine out of the brain, you get these big field potentials that lots and lots of neurons lock to, and now you've lost behavioral flexibility because all the neurons are doing the same thing at the same time. And that giving levodopa back somehow desynchronizes them and allows them to do their own thing. So some of the things that we've been working in the lab on more recently is, one, how does this synchrony happen? So this seems to be a property of this circuit. Well, where does it come from? And there are lots of proposals for where this might happen. The place we've been interested in, so if this is our sort of canonical basal ganglia circuit, usually this chunk of brain called the thalamus is sort of stuck off to the side and sort of regarded as this monomorphic entity. But in fact, it has a lot of different subregions. And so we got interested in this chunk of brain and especially how it connects back to the cortex. So one of the interesting things about this chunk of brain is that these neurons really like to switch between two firing modes. So they have one firing mode where they sort of fire, 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 and another firing mode where they sort of machine gun, just 5 or 6 action potentials in very short order. And what we've seen preliminarily is that when we run our rats in our task, the episodes of bursting tend to be task related. They're not super frequent during wakefulness, but they definitely modulated by the behavior. And what's more, if we take our field potentials, we decompose them into their frequencies, and we look at it over time. So the horizontal axis is time. Vertical axis here is frequency, and the heat map tells you how strong is the oscillation at that frequency. If we trigger our analysis on when these bursts start, we see this big peak right at beta. So sort of suggesting that somehow these two phenomena are linked, although there's a lot of work to do to determine causality. But if it is the case that these cause this and this causes Parkinson's, then maybe this is a therapeutic target. The other thing that we're really interested in our lab is trying to figure out what any of this has to do with really dexterous, skilled limb movements. I have yet to have a Parkinson's patient come into my office and say, you know what, when I nose poke for that reward pellet, it's just not working the way it used to, right? Patients complain about not being able to grab things. And it turns out that we actually have pretty good animal models of reaching. So rats don't do this normally in nature. I mean, they probably can, but it's not, they'll normally grab things with their snout. But you can force them to reach for it, and you can train them up on this. And so what we've done is we've developed an automated version of this where the pellet gets delivered. We can track the paws in real time. We can try to turn neurons off and on in various ways. And then actually use machine vision algorithms to identify where the paw is in both views and then pull back out what its three-dimensional trajectory is. So not only do we want to understand, well, does the rat get worse at reaching if you scramble this part of its brain, but in what way does the rat get worse at reaching when you scramble this part of its brain? Does it make the movement slower, faster, clumsier? And so those are the things we're really going at. So the big questions that we're interested in the lab is, first of all, what do the basal ganglia do? So one of my most humbling experiences as a trainee was going to this big basal ganglia meeting, being all excited. There was this panel with these titans of basal ganglia research, and they were going to talk about what the basal ganglia do. And they all said something different, and they all qualified it with, I don't know if this is right or not, which is a very frustrating way to enter a field. And then the next question is, if we can figure out what the basal ganglia are for, the next question is, well, how do they do it? What are the neural mechanisms across systems that allow them to do this? And how do those go wrong, not just in Parkinson's disease, but how do they go wrong in Tourette's syndrome, in dystonia, in psychiatric disorders that have probably a basal ganglia component, like ADHD? But one of the things that's really exciting about the explosion of DBS, aside from the fact, obviously, that we can take better care of our patients, is that we now have access to human deep brain recordings. There are other ways to get them. So, for example, people going for epilepsy surgery might also get deep brain recordings. But we have the opportunity to take our hypotheses that we generate in animals, see if they happen in humans now. We have the opportunity to generate hypotheses in humans, take them back to the animals where we can do a lot more detailed, fancy manipulations and bounce back and forth between them. And then the last point I want to make is that it's really important to pick your behavioral assay properly. So you can have the fanciest technology in the world, but at the end of the day, you want to correlate it with behavior. And so that's why you need people developing really clever tasks that really tease out the specific neural function that you're trying to go after. And so this is my last sort of meat slide. So because we're at this meeting, and I've already seen a ton of talks where I thought, gee, what could I do with that? Like, this would just be awesome. So there are a lot of new techniques that are coming into the field. So optogenetics, rendering specific neurons light sensitive, because right now you have jumbles and jumbles of neurons of different types in different brain regions, so it would be nice to be able to manipulate them separately. We have all kinds of new circuit tracing techniques. We have new optical recording techniques to selectively record from specific neuronal types. So we drop our electrodes in, we record neurons, but we don't know what kind of neuron we recorded. And in human recordings, we're starting to see multi-site probes. So instead of getting one neuron at a time, you can get several neurons at a time when you do your deep brain recordings. And there are even devices now where we can download field potential recordings from patients that have a fully implanted system. So we can actually do behavior outside that setting. But what do we need? Well, so what's the dream of every sort of systems neuroscientist? It's to have a way that is non-invasive to get cell type specific recordings or manipulations, turn things off or on, that we can do simultaneously in the entire brain on a millisecond or sub-millisecond time scale with single cell or maybe even synaptic resolution. And then once we have all that, so we know what the entire brain's doing all the time, then we need a really smart mathematician to figure out how do we deal with all these really, really complicated data. And so with that, I'm going to finish and thank all the folks in my lab, my mentors, Josh Burke, Roger Albin. By the way, Michael, Roger gets really upset if you call that the classical model. He says that it's classical is a type of music or a type of architecture. But it does not apply to a model of the basal ganglia. And of course, the deep brain stimulation team at Michigan. So at Michigan, you're not allowed to call anything deep brain stimulation or what everyone else in the world calls it. You have to put a maze, not a gold or yellow, a maze M in front of it, and then it can be the Michigan program. So of course, Dr. Patil, Emily Levin is our neurosurgeon at the VA, Dr. Chu is the other neurologist involved in our program. Laura basically makes everything work, as does Kelly, and I'll have to apologize to Wilma. And with that, that's what I have. Thanks.